Hello and welcome back. Today we'll be starting with the plasma proteins. So the plasma proteins we can find in a blood sample. After centrifugation, the blood will be separated into the plasma, the buffy coat, the histopack, and the blood sample down here. So the plasma is the liquid portion of blood, and it's treated with the anticoagulants. It contains water, the plasma proteins, electrolytes, hormones, gases, various metabolic waste. The buffy coat, you can barely see it right here. It's like a little grayer disc. It contains the white blood cells and the platelets. And then histopack, you don't really need to know about it, but in the bottom, here it is. These are the erythrocytes. The serum is the liquid part of the blood after coagulation, and that contains no clotting factors. And there you go. The hematocrit is the red blood cell volume to total blood ratio. The hematocrit is important in the diagnosis of anemias. So the plasma proteins, there's a variety, and they are all synthesized in the liver except for the immunoglobulins. They are glycoproteins. So remember what glycoproteins are. It is a carbohydrate attached to a protein. And polymorphisms happen in all of these proteins. It just means that they are unique to the individual one, and two, it is a per needed modification. So if something like regulatory proteins are like that, so if you needed a gene to be silenced, an enzyme will attach to the protein, and it will change its function due to the binding to the allosteric site. And that same protein could be used when something else, another substrate, binds to it into a different area, and it would change its modification. It will change its function, so instead of silencing the gene, it may activate the gene. Just to reiterate, it is unique to the individual, so they're important to note whenever you are performing blood transfusions. The body may reject them, and the patient might go into shock. So most of the plasma proteins are acute phase proteins, so they are just a as needed basis. Okay, so one of the major players is albumin. We all heard about it, right? Albumin is the most abundant protein in plasma. It's made in the liver. It's soluble in water, and it's negatively charged. That's why it interacts well with water. It is present in the extravascular fluid, the cerebrospinal fluid, the urine, the amniotic fluid, and the interstitial fluid. And it has a role in transport, antioxidation, enzyme activity, and it also contributes to maintaining the oncotic pressure. Oncotic pressure, once again, is the pressure gradient between the blood in the vessels and the blood, or the fluid in the vessel and the fluid within the cells of organs. So these are the clinical aspects. Albumin binds quite well to drugs and affect the pharmacokinetics. And for that reason, we have to be careful about who we administer these drugs to. So sulfonamides release unconjugated bilirubin, and these cannot be given to babies because it will form something called kernicterus. So kernicterus is when the bilirubin, the unconjugated bilirubin, moves from the bloodstream into the brain, and that can be aggravated by the sulfonamides. So in the liver disease or starvation, albumin synthesis decreases, and that causes the edema, specifically abdominal edema, also known as ascites. And albumin can be given to treat the burns and hemorrhage. So why all of that, right? It's because of the ability for albumin to draw water back into the tissue. So that's why they treat burns, and they preserve the blood volume in hemorrhage. Okay, so alpha-1 antitrypsin is part of the alpha-1 globulin. It is a serine protease inhibitor, so it inhibits trypsin along with elastase and protease. And a defect in the genes coding for alpha-1 antitrypsin plays a role in emphysema, because our neutrophils release like elastase normally, and to clear out the lungs, this is a normal maintenance to keep our lungs clean. We do inhale quite a bit of pathogens and dust and dirt on the daily basis, but we have the alpha-1 antitrypsin to inhibit the neutrophils if if there's too many of them, if a signal has gone wrong. So the antitrypsin will prevent the destruction of the alveolar. So in emphysema, the mutation is a glutamate to a lysine, so an acidic amino acid to a basic amino acid. And smoking also inhibits alpha-1 antitrypsin methionine, which inactivates the alpha-1 antitrypsin. And then you can treat this with weekly infusions, IV, of alpha-1 antitrypsin. So the alpha-2 macroglobulins are they play a role in the humoral defense. They're a glycoprotein with 4 identical subunits. It's a pan-protease inhibitor. What does that mean? It just inhibits a lot of proteases, such as trypsin, thrombin, and collagenase. It binds the cytokines, such as PDGF and TCFB, to regulate cell growth and function. And it's increased in nephrotic syndrome. So haptoglobin is an alpha-2 glycoprotein, and its main role is to bind to the free hemoglobin. It's created in the liver, and when it binds to the when it binds to the hemoglobin, it causes it not to be able to pass through the glomeruli, and because of its size, and it prevents the loss of the free hemoglobin into the urine. So what happens when things are defective? In normal anemia, the spleen works, and erythrocytes are degraded in the cell. Erythrocytes are degraded in the spleen, and the concentration of haptoglobin in the blood is increased. So in hemolytic anemia, there's an enzyme dysfunction, and it causes spleen dysfunction and eventually spleen destruction, and the amount of haptoglobin is decreased. So this is an indication to hemolytic anemia. So you treat it with enzyme therapy, also known as supplementation. As you can see here, it plays a role, all of that, spleen, kidney, liver, and it causes the acute hypertension and vascular injury, because the free hemoglobin will cause blockages in the vessels. Okay, ceruloplasmin binds to copper, and it's made in the liver, the serum, ceruloplasmin is made in the liver. That plays a role in transporting copper all over the body systemically. And this is the other version of ceruloplasmin, and it is created by the glial cells in the brain, and it processes iron. So because of that, prions can infiltrate the brain. So copper is an important cofactor. It has many roles in enzyme regulation, activation, signaling, and in low levels it causes Menkes and Wilson's disease, which we'll go through, and in high levels it causes acute or chronic inflammation. So this is the normal route of copper. So it's from like liver, not just starfish, but shellfish in general, nuts and chocolate. So it goes in the stomach, absorbs in the blood, processed into the liver. So Menkes syndrome is a defect in ATP7A gene, and it is a problem with copper transport. So yes, there's a deficiency of copper, but it is a problem with the transportation. So when you eat the copper, normally, you eat it and then it's absorbed and then it's processed in the liver. But instead of being able to be processed by the liver and like delivered to the brain, the skin, and the bones, it's just excreted out. So it is defined by sparse, coarse, twisted hair, kind of like a steel wool, the sagging of the face, and hypotonia. Remember that copper plays a role in collagen synthesis, and that is why the face will sag and the muscles will be atrophied. So children born with Menkes syndrome are failure to thrive, and they have, unfortunately, a deterioration of the nervous system. So the ATP7A protein transports copper from the food and into the enterocytes, which are the intestinal cells. And in the other cells of the body, it will transport the copper from the Golgi and the cell membrane. See, it regulates like intercellular copper levels, and it will cause the low level of copper in the plasma, the brain, and the liver, but paradoxically it will accumulate in the duodenum, the kidney, the spleen, pancreas, and the skeletal muscle. And like I said before, it affects collagen production, and specifically lysyl oxidase, the enzyme that phosphorylates tropocollagen to collagen. So Wilson's disease is also, a deficiency in copper, but this time it is a problem with copper absorption. It's a defect in ATP7B, and it will eventually cause like a liver disease and CNS degeneration. It is defined by the Kaiser-Fleischer rings. Look here. So you can see this quite well just like in a normal physical examination, but to really confirm it, you should use a split lamp to see into the eye. So ATP7B transports copper from the liver to the rest of the body, and it is important for the elimination of excess copper. The defect causes excessive copper deposition as it leaks out of the liver, and you treat this with penicillamine and trientine, which are the copper chelators, and then zinc to block the dietary zinc absorption, because zinc plays a role in copper absorption. So transferrin is a beta-1 globulin, and it transports 2 moles of the ferrous ion per 1 mole of transferrin. It's in the negative acute phase, And iron overload occurs if transferrin is misfolded or the receptors are defected. So here you go. So iron comes, binds, and then this transport molecule takes it into the cell where it dissociates, and then iron can be used, and then it is either exported through the ferroportin, or it's released through the cell surface using the transport. So immunoglobulins are glycoproteins made out of plasma cells, and the complement activation of white blood cells, mast cells, phagocytosis, and apoptosis is what the immunoglobulins are responsible for. And there's several different types. The membrane immunoglobulins have antigen specificity roles, while the secreted immunoglobulins are the effectors of the humoral immunity. So this just kind of like alerts the system, and this is when the system is working actively. So here are the different kinds. They're defined by the 2 light chains and then the 2 heavy chains. And the light chains are bound by the heavy chains. The first 100 amino acids of the N-terminal usually vary in both chains. And the complementary determining regions is where the antigen binds. So around here, here, here. This is the hinge region. And that is rich in proline, cysteine, in the immunoglobulin G and A and D. IgM and IgE lack the hinge, but they have extra CH4 domains instead. So these are the classes we should know. IgM is the main antibody of primary responses. It is a B cell receptor. IgG is the main antibody of secondary responses, so those neutralize toxins. IgA is secreted in the mucus, tears, saliva, and breast milk. IgE is an allergy and anti-parasitic. And IgD is a B cell receptor. All right, thank you for your attention, and good luck studying.